Welcome to part 2 of chapter 21 on the evolution of populations. Here we're going to talk about these forces of change that will change these allele frequencies and really therefore drive evolution. The first one is mutation. This is something we've already talked about. It's fairly straightforward. The idea is if you have a substitution like we show here, or an insertion deletion, is you have a new allele, and that means allele frequencies have therefore changed. Now, a new mutation may not make a huge impact on allele frequencies, but it is a new allele, and if it's potentially selected for, it might become more common in that population. Non-random mating. So remember, Hardy-Weinberg assumption, there has to be random mating for those just alleles to recombine at random in new individuals during fertilization. Self-fertilization, like we see here, that's non-random. So this individual plant is selfing in this example. So that's non-random mating. Here we see an example here with these beetles. They're choosing mates based on the color. This on the left is this is random. This dark colored beetle will mate with any colored other beetle. Where we might see non-random mating is here. The dark colored beetles maybe only mate with other dark colored beetles. So that would be a non-random process. When like individuals tend to mate together, this is called assortive mating. And let's say these dark-colored beetles didn't breed with the other dark-colored beetles. That's called dissortive mating, where they're choosing individuals that look different from them. Now we'll spend a little more time on the next three drivers of evolution here, and we'll start with genetic drift. Now this is the one that probably, when you're first learning about it, you connect to the idea of population size. Yeah, that is important, but more importantly, genetic drift itself is the random fluctuations. So it's random stuff that can happen to a population. It's important for small populations because if you're a small population, you're more likely to be impacted by a random event. Let's say there's a population of 20 chipmunks and there's a landslide and it kills 15 of them. That's a huge impact on that population. But if there's a landslide and you have a population of, let's say, 10,000 chipmunks and it kills 15 of them, that random event is going to have a much smaller impact on that huge population. So that's where the population size comes in. But keep in mind, drift is all about random events. So there's two aspects or two components of this where we see drift having a big role. One is called the founder effect. So again, where we have a small population establishing a new population, few individuals establishing a new population. Something else is called a bottleneck effect, where a population collapses in size and it's down to only a few individuals. And again, smaller population is going to be more susceptible to these random events. So let me demonstrate with this. This is a population genetics simulator. We can start with let's start with an infinite population here. And we've got 5 different populations. We'll just keep the allele frequencies the same. It basically allows us to run this simulation 5 times if we have 5 populations. And we're going to go 400 generations. And I can just click go here. We only have 2 alleles. Allele frequency of A1 is 50%, so A2 is also 50%. Run the simulation. Look, straight across. An infinite population is going to be genetically stable over time. Again, assuming there's no mutation, there's no migration, no selection. Stable over time. So this is going to illustrate drift. Let's create a finite population now. So this is not infinitely huge. We'll still use 5 populations, 400 generations. How many individuals? Let's try 100,000. I'll hit go here. Just got to think about it. All right. So still pretty stable. But look, the lines aren't perfect here. There's some fluctuation. Those are just random chance events that will change allele frequencies. And so let's decrease this now. Let's try 1,000. Population's getting smaller. Or we're looking at groups of species that have a smaller population, however you want to think of it. Now you really start to see when you have a smaller and smaller population, these random events start to change allele frequencies. Let's think of a really impacted population, 100 individuals left. Look, we see really big fluctuations here. Notice in the red line here, this example, the frequency of the A1 allele goes to 100%. That means the other allele, A2, is gone kaput out of that population. It's gone. You've lost that genetic diversity in that population. This is called going to fixation. In this purple one down here, we actually lose the A1 allele. That means the other allele goes to fixation. It becomes the only allele in town. And we actually see that in the green and brown. Those ultimately become fixed over a few hundred generations. Let's take an even more extreme, a population of 10 individuals. So, think about this from a conservation biology standpoint and maintaining genetic diversity. If you only have 10 individuals, that population is seriously imperiled. We click here, it takes less than 50 generations for these alleles to become fixed. So either the A1 or the A2. And that's important because you've lost an allele. In this red population, we lose A1 pretty quickly. In 13 generations, that's gone. And so the link for this is on the slide. You can tinker with this, different population size. You can mess with the relative fitness of these different groups. Think about selection and migration and mutation. There's all sorts of things you can do. But this demonstrates that impact of the small sample size as how it will be impacted by these random events. All right, so where do we see these small populations coming up? One is with this founder effect. So in this example here, we have these individual birds. Say there's phenotypic variability here. You've got yellow birds and red birds. If some red birds, only this handful of individuals end up on this island and they all happen to be red, that is going to reduce the genetic diversity, or not reduce, but the population that's being established there has much less genetic diversity than what was on the mainland over here. So that's called the founder effect. Another place that we see genetic drift having a big impact because of a small population is on something called a population bottleneck. It's called a bottleneck because you can imagine it, well, like a bottleneck here. So if we had a parent population, and here's the allelic diversity here, we've got green, yellow, some orange alleles, and a population crash. So you lose a high percentage of your population, and this is what gets sort of quote unquote passed through the bottleneck. Here are the surviving individuals, even if your population rebounds. So the numbers come back up, but look, there's more green alleles than yellow than there used to be. And there's no orange allele at all. You've lost that genetic diversity. So there's lots of examples of this in conservation. The California elephant seals were at one point reduced to a small population on this island of Guadalupe, and the population's rebounded. So here's the current range, looks good, but because of that bottleneck effect, the genetic diversity is low. What kind of impact can that have? This is a great study on prairie chickens through loss of habitat in specific states. We're using the example of Illinois here. You had a huge bottleneck, and even though the population's rebounded, this has long-lasting effects. So let's look at some of the data here. So we're going to compare Illinois. We have some historical data too from Illinois, 1930s, 1960s to the 1990s here. And we're going to compare that to Kansas and Nebraska, where there were no genetic bottlenecks in these prairie chickens. All right, so population size numbers here we see, okay, big populations. And then 1990s in Illinois, less than 50. There's that bottleneck. So we go look now, what is the number of alleles per locus, per gene, right? The average number. Back in the 1930s to 60s, about 5.2, which isn't dissimilar from Kansas or Nebraska, where you get 5.8 alleles per locus. So they look at multiple genes, they count up the alleles, see how many are for each gene, and then they take an average. That's how you'd figure that out. What's the impact of that? I mean, 3.7 alleles per locus, that doesn't look too bad compared to 5.2. These can have real-world consequences. Look at the percent of hatched eggs in these different groups. 90+% where you had higher allelic diversity, but in these guys where the genetic bottleneck occurred, less than 50% of eggs hatch in these remaining populations. So this has had a big impact on the reproductive output of this remaining population. So again, if you're interested in conservation biology or endangered species, understanding genetic drift, these random effects, and how that affects genetic diversity is really crucial. Here are several examples of organisms where there is a genetic bottleneck, or the population is reduced to such a level that we know we've lost genetic diversity. And that's a real concern. Even if we can get the numbers back up, we're not increasing the genetic diversity. Remember, that takes time through mutations, gene duplications, all those things that we've already talked about. All right, so what are our highlights about genetic drift here? We know genetic drift, the random effects, that's more important in small populations than large. That doesn't mean random events don't happen to large populations. Absolutely they do. It's just they don't have as big of an impact. Allele frequencies change at totally random. We've already mentioned that. Genetic drift can lead to a loss of variation. Those are the examples that we gave. And flip it around, when you're losing alleles, that might mean some alleles that are harmful might become fixed. And fixation, it means that's the only allele left for that gene. So every individual is homozygous for that. And if it's some recessive allele, that could be very bad. All right, so here's a question for you. Which answer is most appropriate here? All right, yeah, there's two things happening here. You've got a small lab population, so that connects back to genetic drift there. And then the way you could sort of work against that is obviously would be increase the population size. But because you've already lost genetic diversity in that population, you really would need to outbreed those flies, get some flies from another lab to do some outbreeding. And that brings us to our next driver of change, which is gene flow. So bringing in the flies from another lab to breed, that would be an example of gene flow. Naturally, this would be, movement of organisms from one population to another, and then they're taking the alleles with them that maybe don't occur in that other population or occur at a different frequency. So how much migration and gene flow is happening might change those allele frequencies. We can look at a simple example here where we have a population, 3 populations, say at time 0, and then over time you have these populations blending together. And so you ultimately just don't have subdivisions anymore. You're just one big population. And in this example that I've just really made up, we've actually lost this one allele, this in population 3, this R allele that occurred here, that got swamped, washed out. In population 1 over here, the frequency of this P allele was 0.3. Now it's 0.4 in the population. So the numbers are changing, and we've even lost some diversity here. It could go the other way, where you might have different allelic diversity in these populations, and if they merge, or you get animals, organisms moving from one place to another, that might introduce new alleles as well. And so the movement of organisms and then introducing alleles to different populations can have a variety of effects. One could be beneficial. Sometimes that might actually have a negative impact, where you have the flow of individuals and genetic diversity from one population to another that might actually sort of be working against local adaptations here. So the example in the text, and this is a really nice example, are these snakes, water snakes in Lake Erie. Now, on the mainland in both Canada and the United States, Ohio and Ontario here, you have snakes that have a very strong banding pattern, and that's favored in terms of, camouflage. But on these islands in Lake Erie, being unbanded is better camouflage. So there would be strong selection to be unbanded in those places. And so much so that you would think, well, eventually all the snakes on these islands are going to be unbanded. You won't see those at all. But migration is happening between these two places from the mainland to the islands. And so you're constantly introducing via gene flow the alleles that determine this bandedness in these snakes. And so if we look on the islands, the degree of banding here, where you go from A is strongly banded to D not banded at all, you still do see more unbanded snakes, and then some with lesser degrees of banding. So overall, they tend toward being less banded, but you're always getting that inflow population from the mainland that's introducing those banded alleles, kind of working against, in this case, local adaptation. Gene flow, as I've mentioned, could improve adaptations to local conditions. The example here is this mosquito Culex pipiens. This is a vector of West Nile virus where we have sprayed for mosquitoes using pesticides. That's, removing those mosquitoes from the population that are not resistant to the pesticides. And then what happens through gene flow and migration is you get mosquitoes that are resistant moving into those areas. So that's a benefit for those mosquitoes. Now you're having new individuals moving in and bringing those alleles for resistance into those regions. All right, let's talk about our last one here, which is selection, which we've talked about a lot already in previous chapters. Just some reminders. Natural selection brings about adaptive evolution. So this is the only one of these forces of evolution where we see adaptive change. Now, remember this slide, variation must exist in the population. If you only have, one phenotype, you're not going to have selection because they're all the same. That variation must be inherited, so it's got to be heritable, and it must result in differences in number of offspring. That has to do with, that's biological fitness, and we'll talk more about that shortly. But we can hit a couple examples of natural selection, thinking about genetic diversity and allelic diversity here. And we'll use the example of owls preying on mice in the Tularora Basin. So you have these different kinds of habitats, these sort of sandy habitats and these kind of volcanic rock type habitats that are darker. And you can really see in this picture here, mice that have a darker colored coat. The owl is going to be less likely to see those against the dark background. And the light colored mice are much more conspicuous, but the situation is flipped when you go into these sandier habitats. The dark colored mice are going to be more conspicuous. And so we could look at the genetic diversity, and if we said, well, allele big D codes for dark fur, we would see selection against or decreasing numbers of that big D allele in these sandy, light-colored, rocky habitats for these dark-colored mice, right? Because they're getting eaten by owls. And then again, it's flipped around when we look at these dark-colored habitats. These light-colored mice are going to be more conspicuous, and therefore you'd see that little d allele decreasing over time as a result of natural selection. So that's how selection can change allele frequency. Now this, seems like an obvious example. There may be more underlying factors here, other genes, other ways, other predators. There may be, we've talked about trade-offs. There may be other consequence of having dark fur against a dark background. And we can dig a little deeper if we want beyond the phenotype, looking at genetics and counting up those alleles for many genes if we want. And that can really help to put this whole picture together, which starts to become more interesting and complex the deeper that we go. Now, this is an important question. Is one of these coat colors optimal? And the answer to that is no. That it's just, you have to look at the scenario, the situation, and compare it to something else, right? That's where that variation is important, right? So it's not all the mice are just dark colored coats, because then natural selection has nothing to act on. And so one of the things we can look at here is these comparison. We're comparing different groups or different phenotypes, genotypes, and this is all about relative fitness, because you have to compare one group to another. So let's use peppered moths as an example of relative fitness, where we see different environmental conditions and differences in fitness of a dark colored moth, like we see here on the left, or this lighter colored moth. In this case, it's blending in nicely with its background. So that's the scenario. So let's start with a few details here. If we look at these moths, these variants, this variation, phenotypic variation, is heritable. If an individual has one of these A2 alleles, they'll have this black morphotype. If they're homozygous for the A1 allele, you have this light-colored phenotype. So the black is dominant and gray is recessive. Now, in this condition, with this background, these dark-colored moths are going to be more conspicuous, and this bird here is, munching down on the black-colored moths. And so this is a classic example of natural selection. But there's also a historical context which is interesting. So here we see different environments where the black over here on the right is less conspicuous. It's more conspicuous on the left. On these trees that these moths are found on, there are lichens on the surface that give them this light-colored appearance. And again, the light-colored moths would be much more camouflaged in that case. And then during the 1850s, this is the Industrial Revolution, pollution was much more common, and this basically killed a lot of the lichen on these trees, so that you don't get that lighter colored look to it. Also, then the bark is a little bit darker underneath the lichen. Here's where those black moths are going to be favored. So this is kind of interesting where we've seen this over time. And then when we start to recognize that this as an environmental problem, we start to clean up some of this air pollution, and we see this trend reverse. So if we look back here from 1959 to up to 1995 here, the percentage of the black moths, the melanistic moths, decreases because we start to see more of those lighter colored trees as those lichens recover. Pollution is less. There's less of the soot on the trees. And so we see fewer of the black-colored moths. So this is just kind of an interesting example of natural selection, but we're going to look at it in a context of biological fitness here as well. But first, a question. In a population of animals, the most fit individual would be what? Right. It's all about production of offspring. So I like to show this slide in thinking about fitness. You might think about sort of athleticism, or this, powerful shark, or a tree that's big and tall and strong. But, this family here and these, great grandparents or great grandparents here in the middle, these are the most fit individuals potentially in these images. The trees, maybe we're not sure about, in the shark. But it's all about offspring production. So biological fitness is all about relative fitness, comparing one group to another. You've probably heard the term survival of the fittest, which we sometimes say is misleading because it implies this competition between individuals. And that doesn't mean competition doesn't happen, but ultimately what matters from a biological fitness perspective is the contribution that an individual makes to the gene pool of that next generation. So if one individual has 30 offspring, another individual has 2, that individual that has 30 has had a greater impact on that next generation. Now, why might an individual have 30 offspring versus 2? That might be because they have certain traits that allow more opportunities for mating, or they just live longer so they can reproduce more, and so on. All right, so let's loop back to our moths here and look at how we might measure relative fitness. So we're going to take some experimental studies here by Bernard Kettlewell, and we can put this in a context of a hypothesis and say, well, if the color of the moth compared to its background influences predation, which, we think that's what's happening, but we need to test this. If a moth doesn't match their background, they should have lower relative fitness than those that do match their background. So we're making a comparison. That's really important. We're comparing two different phenotypes here. So we can do that. We can plug in the data that we gather here. So how do we know what the survival is? Well, you can do a mark recapture study. So we'll capture, like, hey, here we got 406 dark colored moths collected from an unpolluted forest. We tag them, mark them somehow, and then we go back later and we recapture 19 of them. And that gives us a sense of the percent survival. We're going to say 4.7. Light colored moths, same thing. We get about 393, and when we recapture 54, the percent survival here is much more, 13.7. So we can now figure out, well, what's the relative fitness of a dark versus light colored moth in an unpolluted forest? We take the bigger number here as the baseline. You're always choosing the bigger number first. And we can say, well, the relative fitness of light moths in a light forest is 1. That sets the standard, so to speak. And then any other groups here, it's just one group, with the lower percent survival, we would calculate what that number is, 4.7 divided by 13.7, our baseline. So lower relative fitness. We can just run through the same thing in our polluted forests, the survival of dark moths versus light moths. And we see, again, the scenario is flipped here. The relative fitness of the dark moths is higher here, so that sets our baseline, 27.5. And then we can plug in these relative fitness numbers into a table, if that's helpful, and make more direct comparisons here. And what we see is the relative fitness of dark moths in unpolluted forests is less than the light moths in the polluted forests. Another way to think about this is this idea that means selection is much stronger against black moths here in the unpolluted forests. Birds are seeing them more readily, perhaps. And we can express that as a selection coefficient. And all we're doing here is subtracting 1 minus 3.4 or 1 minus 0.48. And what we see, the selection coefficient is much higher. So that means selection stronger in these unpolluted environments versus polluted. Again, we're always comparing one thing to another. Okay, so what scenario or scenarios can explain these data? All right, so yeah, any of those scenarios that I presented could explain it. It's easy to jump to predation by those birds because that's kind of how we introduced the idea. But just thinking with an open mind, you couldn't rule out those other scenarios that were presented. You'd want to do more research to figure out exactly what was happening. There's a difference in relative fitness, and then figuring out why that is would take some more study. Okay, so let's look now. What's the trajectory of these changes that might happen in a population? We can categorize them in a few different ways. If we're looking at continuous variation, say like height, or in this case, this mouse fur color where there's a gradient, it's not just say black and white, for example. We have a distribution of that trait in the original population, and we might see 3 different things that could happen. One is called directional selection, where in this scenario, you're starting to see more and more dark colored mice over time or over generations. Disruptive selection, you're seeing the extremes becoming more common and those in the middle getting less common. And then stabilizing selection is kind of a narrowing. You're seeing an elimination of the extremes where you're losing over time the really light colored mice and the really dark colored mice and favoring these intermediate types. And so here's some other examples of, say, directional selection. And this is an experimental study that illustrates this, where we have fruit flies and we'll put them into say a tube, and there's a light at one end. And if the flies work their way down the tube toward that light, we're going to take them out, squash them, remove them from the population. So you're selecting for individuals that don't tend toward the light. And over multiple generations, what do you see? You see a decrease in the remaining populations to work their way toward that light. Because you're selecting against individuals that migrate toward the light. And this is a directional selection. You're shifting the population to those that stray away from light. Disruptive selection. The example here would be, say there's only two kinds of food, seeds for these birds to eat. There's either big seeds or little seeds, and there's no intermediately sized seeds. And so that will favor individuals that have either a big beak that can take down those big seeds or a little beak to take down those small seeds. But individuals with the intermediate sized beaks, they are going to have reduced relative fitness because they're not so great at eating either big seeds or small seeds. Here's another example of disruptive selection. We have flies that feed on apples versus flies that feed on hawthorns. And so before apples were introduced into North America, that's where this example is happening, these were flies that were just feeding on hawthorns. But as apples are introduced, you see some of those starting to take advantage of apples, and so much so that by, say, 1991 here, we actually have different populations of flies. So we're starting to see this separation and really on the way to speciation of these two different flies. Stabilizing selection is removing those extremes from the distribution. And the example that I give here is human birth weight. So if we look at the bars here, these are the births in the population relative to size. And then the line here shows infant mortality. So really small babies, usually premature, or really big babies, we start to see increased infant mortality. And we see fewer of those births. We see a lot of births kind of in the middle, sort of sweet spot in terms of size and lower infant mortality. So this has this narrowing of the distribution. All right, so here's a question for you, and pick the answer that fits best. All right, yeah, this one is right out of the last example. This is stabilizing selection. So sort of you have a narrow range of numbers of eggs that seem to give the best relative fitness. Let's talk about another trend that we see called balancing selection. This looks a lot like stabilizing selection, but we're really looking at maintaining alleles in a population and really pushing toward like more heterozygous individuals. Whereas the stabilizing selection, that's more narrowing selection, that there might overlap a bit with balancing selection, but we're really eliminating extremes from that distribution. So they look very similar, but there are some subtle differences. The oft used example of balancing selection is sickle cell anemia. This is also an example of what's called heterozygote advantage. We've talked about sickle cell anemia before. If you have that sickle cell allele, we're going to call it S. If you're homozygous for it, it's usually lethal. If you're homozygous for normal hemoglobin, you don't have sickle cell anemia. That sounds great. So why would this allele be maintained in the population at all? It's because of malaria. So if you are heterozygous for this S, so be SH, somebody who has the SH genotype, you have some resistance to malaria. So that's really driving that maintenance of that allele in the population. And so again, thinking about stabilizing versus balancing selection, we're pushing toward heterozygosity. So we're maintaining those multiple alleles in the population, and that's a good example of balancing selection. So individuals that are heterozygous, they don't have sickle cell anemia, and they're less susceptible to malaria. So that's why that's an advantage, even though homozygous individuals have an extreme disadvantage. And this here we see these sickled cells, and I'll just point out this is malaria. This is Plasmodium falciparum. That's the worst form, really, of malaria here living in a red blood cell. So another kind of balancing selection that we see is called frequency-dependent selection. And that's just as it sounds. It depends on how common a certain phenotype is, determining what sort of selection that we see. What are we going to see will happen? And there's two kinds. of frequency dependent selection. One is positive frequency dependence. What you see in positive is the fitness of a phenotype will increase as it becomes more common. So that's the positive side of this. Another way to think of it is, like we see in this example here, is these, say, oddballs stand out. So here's this little fish that kind of stands out against the background of all these other fish, and the predator is cued in on that. This predator is gonna go after this guy that stands out. So being the more common type is a benefit. The other kind of frequency-dependent selection is negative frequency-dependent selection. This is just the opposite of positive. Is as a genotype is less common, you see greater fitness. And so the example we have here are these water beetles. And here we have fish preying on these water beetles. We have different colors of water beetles. As a certain color becomes more common, the fish cue in on that. They're not going after the oddballs like we saw in the last example. They're going after the ones that are most common, which is a good strategy, right? Because if you go after the ones that are most common, you get good at catching those. That's great because there's lots of them. And so that benefits the beetles that are rare. So if you're a light brown beetle and the fish are going after the dark brown beetles, your relative fitness could increase because predation pressure has dropped down. Now that might flip around over time as now the light colored ones become more common and the dark colored ones are less common. Now they benefit from that negative frequency dependent selection. And so we can see that really in action here. This in the past has been called oscillating selection, but it's really an example of negative frequency dependent selection, where we have these scale-eating fish here that come and nip away at the scales of this target fish. And the most common type, let's say these left-mouthed fish are really common. This fish that's being picked on is going to start to really pay attention to its right side here against this left-mouthed fish. And of course, as they do, then now these right-mouthed fish have the sort of an advantage, right? They're the less common type. They have an advantage now, and they'll start nipping on this side. Now, of course, now the situation will flip around. Now the target fish is now paying more attention to its other side. And so we see this changing, oscillating over time as a result of this negative frequency dependence selection. Another kind of selection is called sexual selection, and this connects to the idea of non-random mating. So this might tie in with something called sexual dimorphism. Dimorphism is where you see two different morphologies. When we see sexual dimorphism, we might see where males and females of a species look different. That's what that's all about. So in this case, with these photos here, we see males of different species with these different, sometimes elaborate displays that are potentially meant to attract a mate. There's a couple of kinds of sexual selection. One's called intra-sexual selection. That means within a sex. And so where we see often males competing for mates of the opposite sex. Intersexual selection, so that's between the sexes, is where we see females choosing mates. Again, typically the females are choosing the mates, and the males often have these showy characteristics like this peacock or this little guppy here that are meant to attract mates. All right. So sexual dimorphism is a result of what? Right. This is all about intersexual selection. Rather than intra. So just making sure understand the differences between those terms that sound very similar is key. All right, so another idea with sexual selection is something called the good genes hypothesis. And this relates to the concept that a female is choosing a mate based on some of these like showy characteristics, or in this example here, we'll describe calls by these individuals, these frogs, male frogs, and they're choosing those mates because of those outward characteristics, but those outward characteristics represent quote unquote good genes. And so how do we know that those are good genes? We have to think back to relative fitness, right? So which, matings would produce more individuals and better survival in the offspring? Those are the things that matter from an evolutionary perspective and for the next generation. So in this study, we've got these short calling SC males and long calling males, LC males. Now, the idea is, well, these long calling males are potentially have better genes. They're going to attract more females. And so you can set this up experimentally. We're going to take eggs from a single female. So we're taking that out as a variable, just one female, eggs from her, split those up. And we'll fertilize those with a short calling male sperm versus long calling male sperm. And then we'll look at the results. And what do we see here? Larval survival, and we experiment done over 2 different years. The long calling survival, the offspring were better. NSD means no significant difference. So, okay, it's kind of a wash in 1996, but larval growth, not different, but long calling males offspring better. Time to metamorphosis, both years, quicker metamorphosis, so long calling individuals better. So all these measures, potential measures of fitness, seem to suggest that, yeah, these longer calling males, it is a good representation of their good genes because their offspring have better survival parameters as a result. Now, this calling could be a trade-off. And this brings up this idea that natural selection doesn't fashion some perfect organisms. And this connects to relative fitness too, right? You're not creating some optimal organism. It is just better than what else happens to be there, right? A potential outcome of any of these scenarios would be that an organism goes extinct, right? You're not moving toward some optimal situation. And it's because selection can only act on what's already there. Mutations may occur, gene duplications, these things can occur, but selection can only act on what happens to be there. And then, therefore, is limited to whatever historical constraints, the genetic, environmental, all these things potentially connected. And adaptations are often compromises. So think of the frog calling. Like, okay, great. The frogs that have longer calls, maybe better at attracting mates. That's some reflection of quote unquote good genes, but longer calling frogs might also attract more predators. This is just a great image, this bat swooping down to eat this frog that's sitting out here calling for mates. And so all of these things potentially come together, chance, selection, the environment, all these things interact and give you the diversity of life that we have today. But there's not some ideal scenario. A good example of a trade-off is one we've heard about many times this semester is the sickle cell trait, where you have this potentially negative disease, but individuals that are heterozygous have resistance to malaria. This is a trade-off. There's a compromise here. I love these cartoons. Adaptation doesn't involve trying. So this, beavers, just wishing for a chainsaw. That doesn't happen. You got to work with what you have. Natural selection works on what's already there, the variation that's there. And natural selection doesn't, quote unquote, give creatures, organisms what they need, right? Again, you just can work within whatever the constraints you have. There are also limits to selection based on all these things that we've talked about. And I like this example here. This is an artificial selection example where we look at winning times for Kentucky Derby winners. If you're not familiar, Kentucky Derby is a horse race. And what you see over time from 1910-ish or so, here are the winning times, 125 seconds-ish. We see that increase a little bit and then decrease. So horse breeders are breeding for faster and faster horses, but you reach a point, a horse just can't go any faster. And so we see it level off and kind of oscillate here. You can't just breed a faster and faster horse at this point. There are limits based on the physiology and anatomy of these animals. All right. We'll see you next time.